name is Pollock, and I'm staff scientist at the PIPOXS beamline at Chess. My role is to help users both plan and conduct their experiments, and also to process, analyze, and interpret the data that they get from those experiments. So PIPOXS stands for the Photon In, Photon Out X-ray spectroscopy beamline. And as you might be able to guess from that name, what we focus on at PIPOXS is studying interesting chemical systems using X-ray spectroscopy. So what I mean by that is we generally take a sample and we hit it with high energy X-ray photons. And in one flavor of X-ray spectroscopy called X-ray absorption spectroscopy, what we do is we look at the energies that that sample absorbs when we hit it with X-ray photons. From that, we can learn things about what the geometry is of that atom that's absorbing, what its oxidation state is, and also how long its chemical bonds are. Now, from the name, X-ray absorption, that's the photon in aspect of PIPOXS. Another flavor of X-ray spectroscopy is called X-ray emission spectroscopy. And what that involves is once we've excited those electrons in the sample, if you wait a little bit, the sample will then give off or emit X-rays of different energies. And so if we look at the energies of those X-rays that get emitted, we can learn different things about the sample that we have in the beam. We can learn things like how many ligands are bonded to that atom, what are their identity, and what is the spin state of the atom that's absorbed those photons. Now, from the name there, it's X-ray emission. That's the photon out aspect of PIPOXS. So together we have the photon in, photon out, X-ray spectroscopy beamline. The main goal of the PIPOXS beamline is to investigate chemical catalysts. And so a catalyst is a chemical species that speeds up a chemical reaction without being consumed in that reaction. As you might imagine, catalysts are quite common both in nature and also in man-made processes. So they're involved in everything from synthesizing DNA in your cells to producing industrial fertilizer that helps to feed billions of people. So they're very useful substances. But unfortunately, in a lot of cases, we don't really know how they work on a molecular level. And that poses a problem because if you ever want to improve how a catalyst works or design a new one, you really need to know how the existing ones function in order to design better ones from that. And so that's where PIPOXS comes in. Our goal is to be able to study chemical catalysts, ideally under operating conditions, so that we know how they function, how their structures might change during a chemical reaction, so that we can then learn from that and design new and better catalysts from what we currently have. As I said, catalysts are involved in all kinds of different processes. And so when we designed the PIPOXS beamline, we really had to make that a very flexible end station, since you have to be able to look at everything from heterogeneous catalysts, like on industrial scales, all the way down to the enzymes in your cells. So we have the ability to look at a wide variety of elements in a wide variety of environments to answer a wide variety of chemical questions. The PIPOXS beamline was designed to be able to look at a wide variety of samples in a wide variety of different environments. And so that flexibility comes from really a number of different factors that we built in. The first is the energy range that we can use to hit our samples with. So we can reach elements all the way from calcium all the way down to uranium. So almost the entire periodic table is accessible to us. We also have the ability to look at a wide variety of environments. So that could be samples that are a liquid in solution, or it could be an electrochemical cell that has applied voltage, or it could be a frozen sample that's at 10 degrees Kelvin. So the environment that the sample is in really doesn't limit what we can look at. Another thing that we have that a lot of beamlines don't necessarily is we have access to several different spectrometers that let us do techniques that you can't necessarily do with all other beamlines. Some of these spectrometers are unique only to Chess. So we can do both X-ray absorption, X-ray emission, and we can also combine those techniques into more complicated two-dimensional methods so that we can really tailor the experiment to help the user answer whatever chemical question they have in the best way that they can. And the last thing that we can tune is we can tune the size of our beam. So we can tune all the way from about a 1 by 2 millimeter beam, so a very big beam, to hit if you have a very big sample, or we can focus that down to about 100 by 200 microns. If you have a small sample, you need a very high flux density, we can tune that as well. Some of the really interesting experiments we've done lately are looking at electrochemical materials. So these could be things that you put into a battery or a fuel cell, And when you either store or release charge, they undergo some kind of a chemical reaction. And in a lot of cases, the people who make these materials don't necessarily know what the chemistry is that's happening in them. So you often have many different metals, some nonmetals, and when you charge and discharge, we don't really know where that charge goes. Using X-ray absorption and emission spectroscopy, though, you can monitor the oxidation states of the different elements in those samples, and because these are inherently element selective, you can look at each one sequentially, and you can see where the charge is coming from and going to as the material goes through its charge and discharge cycles. So you can really learn information that is difficult or impossible to learn without using these X-ray tools. Remote access has actually been going really well at PIPOXS. So thanks to a really dedicated effort by the IT and technical staff at Chess, we've managed to get the Beamline into a state where a user can sit at home and they can log into the station computer and they can see and control just about everything that they could if they were sitting here physically at the station. And our users have actually been really enthusiastic about trying this out. So we've had samples come from as far away as Germany for them to be able to do these remote experiments. And because of that, we really have been able to keep doing exciting science even while we have these restrictions in place because of COVID. And looking forward, I think that particularly for new user groups, the ability to do remote experiments really lowers the barrier to doing synchrotron experiments. And so even once the restrictions from COVID are lifted, I think remote operations will remain an important part of our user program simply because they allow more people to do these experiments that wouldn't necessarily be able to. Because we investigate such a wide range of systems at PIPOXS, there's really a number of ways that this Beamlines research can have a broader impact on society. Probably the most obvious one would be on the energy economy. So we have a lot of research that goes into electrochemical systems, fuel cells, batteries, different electrochemical catalysis. And so what this research can do is it can change the way that we both generate and store electrical power, can make those processes more efficient. There's also research going on that could change the types of materials involved in these systems. So right now they use a lot of both precious and expensive metals and also some hazardous materials. And so with the research that we're doing, we're hoping to be able to replace those with more abundant, cheaper, and more environmentally friendly materials. So if that research comes to fruition, it can have a big impact on how we use energy as a society. We also do quite a bit of research on biological systems, looking at how enzymes function. And so this can have impacts both on just fundamental knowledge, knowing how nature carries out chemical reactions, to drug design, because if you know how enzymes function, you can potentially design drugs to target those enzymes. And also, a lot of these enzymes in nature do very complex, challenging chemical reactions, things that we can't currently do in the lab. And so chemists can then use what we learned about those catalysts as inspiration to design catalysts that can do those challenging transformations in the lab to mimic what nature does. We're always trying to adapt and approach new problems and new challenges at PIPOXS. So right now we're involved in designing a couple new sample environments. So we have one that will enable us much better temperature control over our samples. And we have another one that will enable us to look at heterogeneous catalysts as they do their chemical reactions, which is something we can't currently do. We also are working on a project that will change the way that we do energy scans at the Beamline. So right now it takes us maybe 5-10 minutes to do an energy scan. With this new technology implemented, we'll be able to cut that down to about 30 seconds. So we'll save our users quite a lot of time and we'll be able to do experiments that we just frankly don't have time to do anymore. Those are kind of the big projects that are in flight. And we're pretty much always just trying to push the envelope in terms of how low of a concentration can we measure, how quickly can we scan, and how low of a signal can we actually measure. So we're constantly trying to push the boundaries of what's possible. My favorite thing about working at a Chess Beamline is the diversity of science I get to be a part of. So because this is such a flexible Beamline, I'm always encountering new challenges and new problems and new science. So 1 week I could be working on a metalloenzyme, the next I could be working with the user on a fuel cell component, and the next I could be looking at a small molecule catalyst. So there's always something new, there's always something exciting going on, and I'm always learning new things at this Beamline. So it's a fun place to be doing science.